Moving on, we can now start our discussion of protein structure. And we'll start at the most basic level, which is the primary structure. As we saw earlier, the primary structure denotes the amino acid sequence of protein chains. And therefore, to understand it, we need to understand amino acids first. So here, we can see the chemical structure of an amino acid. And as the name implies, all amino acids contain an amino group, which is positively charged in its free state, and a carboxyl or acid group, which is negatively charged. Both groups are attached to a central carbon atom, which we called C-alpha or alpha carbon. And therefore, the amino and the carboxyl group are referred to as alpha amino and alpha carboxyl. Now we can see that besides these two groups, the alpha carbon is also attached to a hydrogen atom and to a fourth group, which is called the side chain. And the fact is that all amino acids have the same carbon alpha, alpha carboxyl, and alpha amino groups, as well as the hydrogen. But the fourth group, the side chain, is what distinguishes them from one another. And the reason it is called the side chain, because it protrudes from these two groups, which we call the backbone. And the reason we call them the backbone, because as we'll see later, these groups, the alpha carboxyl and alpha amino, are the two groups that form the attachment between different amino acids to form the protein chain. And all three groups actually form the backbone of the chain. That's why we call them the backbone. And therefore, the fourth group is called the side chain because it protrudes sideways from the backbone. So naturally, we'll, in our discussion of amino acids, we'll focus on the side chain because, as I said, it distinguishes between amino acids. But before that, I'd like to say a few words on a general feature of molecules and amino acids specifically, which is called the molecular configuration. And this feature denotes the relative position of atoms and groups around a central atom, which is asymmetric or chiral. And it means when we refer to an atom as asymmetric or chiral, we mean that it is bound to different groups. In this case, we have the carbon atom, which in amino acids is the alpha carbon. And as we saw, it is bound to 4 different groups. So it is asymmetric or chiral. And when we have a molecule with a chiral center like that, the spatial distribution of the groups and atoms around this carbon is called the configuration of the molecule. When we have a single chiral center, like here, the molecule can appear in 2 different configurations, which are mirror images of one another, as we can see here. This means that we cannot, if we try to put, for example, these configurations on the other one, they don't overlap because they are mirror images. And therefore, there are 2 different configurations of the same molecule, which has the same atoms, the same chemical composition. So to distinguish between these 2 configurations, we can use 2 methods. The first one is called the absolute configuration, and there's a way to calculate it. We won't get into that. This method is used mainly by chemists, and the 2 configurations are denoted as S and R. Biochemists use another method, which is also the methods that we will use, which refers to the 2 configurations as L and D. And there is some relationship between L and S and D and R, but they're not always the same. So the second method is called relative configurations, and it is called relative because it was first determined for a specific molecule called glyceraldehyde, and it was then extended to other molecules according to their relation to this glyceraldehyde. But that's not important for our case. We'll just use this method. So why is this feature, why is the molecular configuration so important? It is important because it affects processes of molecular recognition. For example, if we have a binding site in a protein, which binds a ligand or a substrate, in the case of enzymes, as we said, the binding is specific because the binding site is compatible both geometrically and electrically to the properties of the ligand or the substrate. And the geometric compatibility depends on the configuration of the amino acids in the binding site. If they have a certain configuration, they can recognize a certain ligand. But if the configuration changes from, for example, from L to D, they won't be able to recognize this ligand. So most molecular recognition processes in biology are configuration dependent. And indeed, when we look at amino acids, we see that nearly all of them, all amino acids that appear naturally in proteins have the L configuration. We can hardly see any amino acids with the opposite, the D configuration. Here we can see the two configurations. And again, when we look at isolated amino acids in nature, we can find both configurations. But those that appear in proteins usually have the L configuration. And the reason for that is that the system that builds, that integrates amino acids into protein chain, this system is called the ribosomes, has evolved to recognize only the L type of amino acids. But as is usually the case in biology, when we have a rule, we also observe in some cases the exception. And this is what happens here as well in our case. We can find amino acids with the D configuration in certain proteins and peptides, and we can find them in two instances. In prokaryotes like bacteria, we can find them in certain peptides, particularly in antibiotics. And we have an example here of the antibiotic valinomycin, which has a unit that repeats itself 3 times. And when we focus on this unit, we can see that it contains 2 valine amino acids. One of them has the L configuration, which is the normal one, but the other one has the D configuration. And the way the amino acids appear in prokaryotes is that these simple organisms, besides the ribosomes, which are, as I said before, there are the machines or systems that build proteins, they also have non-ribosomal systems, which are enzymes. And these enzymes know how to, in some cases, know how to recognize amino acids with the D configuration and attach them to others to form peptides. And such systems, because they are not as efficient as ribosomes, they cannot create whole proteins, but only short peptides. So this is one instance. The other instance where we find amino acids with the D configurations is in eukaryotes. And then we can find these amino acids both in proteins and peptides, but they appear in a very different way. Eukaryotes don't have non-ribosomal systems. They can only build peptides and proteins using ribosomes. And therefore, all proteins and peptides constructed in eukaryotes are constructed with L amino acids. But after the proteins or peptides are constructed, in some cases, certain enzymes can change some amino acids from an L configuration to a D configuration. This is called post-translational modification because it happens after the protein is translated, after it is formed.